Okay, this is a lovely question. This kind of looks probably a little bit more like the kind of questions you're going to get in the exam. It's got quite a lot of information. It's got a lovely picture. It's got sort of nice long questions. It goes on for 2 pages. It's got another lovely picture. Fantastic, and some more information. So this is pretty much, I think, like the kind of questions you're going to get when you look at last year's papers. So we need to read this properly then. When HIV, the virus which can cause AIDS, infects a cell, the enzyme reverse transcriptase. So already I'm now thinking this is the one we'll be using mRNA template to make DNA, which is found in the enzyme reverse transcriptase, which is found in this type of virus, copies the single RNA strand of the virus into a double strand of viral DNA. Oh, so I was right then. We only get single-stranded DNA, make it into double-stranded DNA. That's what it's telling us there. Viral DNA is then joined into the host chromosomal DNA. So here, what does that word join mean? It means spliced. We're talking splicing. We're therefore talking ligase. Into the host chromosomal DNA, which allows the host cellular process to make multiple copies of the viral DNA. There's been much scientific interest in the structure of reverse transcriptase, and we've got a simplified diagram. So I'm looking at the diagram, and it's an enzyme. So I'm thinking enzyme, I'm looking at a protein. So what can I see that makes me think that that's a protein? I can see an alpha helix here. It's all folded up. It's probably held with some hydrogen bonds. It's probably got a few bonds sort of, between things. So we might be talking about R groups here. Okay, let's have a look at the questions then. Name the substrate of this enzyme. Reverse transcriptase copies the single RNA strand. So the substrate is going to be RNA. That's going to be used as a template. We're going to join our complementary nucleotides in the right order. Name the region on the enzyme where the substrate binds. So this is synoptic, isn't it? So name the region, active site. Super. What evidence from the diagram? I'm going to use something I can see on the diagram. Shows that the enzyme has a secondary and a tertiary structure. So secondary structure is there. There's only 2 types of secondary structure. There's alpha helices and beta pleated sheets. I can't see anything that's really zigzaggy there. So I'm just going to talk alpha helix because there's loads of those. There's one there, one there, one there, one there. Loads of them. There are alpha helices on the diagram. That's what secondary means. And then we want to do tertiary structure. So what's the evidence that it's tertiary? The tertiary structure is secondary structure is further folded, folded into a 3D globular protein held by R group interactions. Now, they've only given me 2 lines into I can't spell interactions. But nevertheless, I know lots about the tertiary structure, and I don't know what's on the mark scheme. So further folded might be a mark, globular protein might be a mark, R group interactions might be a mark, but I'm going to give an example. For example, disulfide bridges. I'm not going to give loads, I'll give 2. Disulfide bridges is my favourite, and ionic interactions. I'm not putting hydrogen bonds because they're holding alpha helices, so they might not allow that twice. And again, that's a bit of synoptic there for you. Explain. So this means give reasons. So I'm going to try and use the word because effectively. Why the enzyme is referred to as reverse transcriptase. So it is called reverse transcriptase because it uses mRNA as a template to make and I'm going to get this word complementary in as well, to make a complementary DNA molecule. And I think that might hit my 2 marks, so one for using mRNA as a template and one making DNA, and then complementary. And I could have written more detail. I could have said the complementary bases to RNA are used to and join together with condensation reactions. I could have written something like that, and I think that will probably be enough. Okay, when DNA is made in this way, mistakes are made, oh dear, and as many as 1 in 2,000 nucleotides might be incorrect. What will be the result of those errors and the importance to the scientists who are trying to produce a vaccine? Okay, so if the nucleotides are incorrect, and then I want to say what will be the result of that, then the base sequence and therefore the codons will be different. Different consequence of that, different amino acids are inserted into protein. So remember, we're not making amino acids in protein synthesis. So a different viral protein is made. Okay, so what's the significance of the viral protein? So vaccines are using those proteins as antigens. So the viral proteins, remember the protein code of a virus, are antigens, and if they change, antibodies will not recognise them. So this is oh, I can't spell. This is something that we came across when we did membrane structure, so a little tiny bit of synoptic in there about antigens. So, moving on. There are combinations of drugs which can be used to treat HIV. One group of these drugs are non-competitive inhibitors to reverse transcriptase. They're acting on the enzyme non-competitively. Describe, AO1 at last. the mode of action of such a drug. So again, a little bit of synoptic. So a non-competitive inhibitor. And the reason for writing full sentences is that while I'm writing a non-competitive inhibitor, my brain is actually working in the background thinking, yeah, what does that do then? Attaches to the enzyme at an allosteric site. And again, you know, if you can't remember the term allosteric, you could put not at the active site, and that would be okay. The drug changes the shape of the active site. So the substrate cannot fit in. I want to get this word complementary in as again. So cannot fit in because, I've got an explain word in there, because the active site is no longer a complementary shape. I'm still not quite filled in my 3-mark thing, but I think possibly I might have written enough there. I could perhaps put no enzyme substrate complexes can form. Frantically dredging back in my memory for what's on the core. So no enzyme substrate complexes can form. Okay. Phew. Right, turn over. 2. Build its viral DNA. HIV uses nucleotides from the cytoplasm. Okay. Cheeky, isn't it? Takes the host cell's nucleotides. Some drugs are made from modified nucleotides. When these are incorporated into the growing DNA chain, the next DNA nucleotide cannot be added, and the newly made DNA strand is terminated early. Now I'm thinking here, we did that thing on Sanger sequencing, so perhaps they're sort of using that kind of technology. Okay, so I'm going to look at here. So this looks like a condensation reaction. So we've got template strand, base, and then we're condensing together. That's the sugar, that's the phosphate. And then here we've got phosphate, sugar, modified nucleotide. Okay, so now why can't it be added on? So the phosphate looks okay. CH2, yeah, pentagon, oxygen base. So there's just, look at this end, that's got an OH and that's got an N3. So modified nucleotide, that's the one that stops it. Yeah, okay. So describe how deoxyribose sugar joins to the phosphate group. So here I can see water being eliminated. So this is a condensation reaction. 2 molecules are joined by a condensation reaction. With the elimination of water. Might come back to that. That seems to be quite a lot of just seems to be a lot of space to write that. And I might need to say it's the two OH groups reacting. Let me just show, I'm going to put that. This reaction happens between the two OH groups. Okay. State why another DNA nucleotide cannot be added after the modified nucleotide. Okay, so my modified nucleotide. The modified nucleotide, let's see how many marks I'm in for, 2. So the modified nucleotide has an N3 group and not an OH group. So what? So it cannot how would you put it? Undergo a condensation reaction because there is no hydrogen or oxygen present. Okay, it wasn't as bad as I thought it was going to be. There are other enzymes which are used by our other enzymes. So I'm already now thinking DNA polymerase, ligase, restriction endonucleases, all the other enzymes we've come across. It's used by the virus to replicate and disperse itself from the host cell. Inhibition of these enzymes can be used to treat HIV infection. The general name of the enzymes used to hydrolyze a peptide bond to remove a group of amino acids from the end. Oh, that's important, the end. So we've got endopeptidases, join them, break them down in the middle. So the other ones are exopeptidases. And again, I think you get away with peptidases, you potentially get away with protease. I don't think you get away with, because it's the general name. So if you named an exopeptidase, for example, trypsin, then I don't think you get it. So general name used to join the DNA strand into the chromosomal DNA. So this is our, I'm just going to go back here, because I think I did stuff here, didn't I? And I said viral DNA joined into host chromosomal DNA, and I put spliced ligase. So I'm thinking used to join the DNA strand into the chromosomal DNA, joining two sorts of DNA together, DNA ligase. Nearly at the end of this. The percentage of each of the 4 types of nucleotide found in the RNA strand is shown in the table. Complete the table to show the percentage of each type of nucleotide in the single complementary DNA strand. Okay, so how are we going to do this? Nucleotide in viral RNA, percentage nucleotide. Nucleotide in single complementary, so I need to name, so these are names. We've got percentages, I need to name them. So cytosine pairs to guanine, and I need to be able to spell these. I need to print them. Guanine pairs to cytosine. Uracil on RNA. Now uracil replaces thymine, is what I'm thinking. So uracil is equivalent to thymine, and A pairs to T, so that's adenine. And adenine pairs pairs to thymine. And that's okay because I've got my DNA bases now, G, C, A, T. And remember, you know, cars in the garage, apples under the tree. So we've got guanine. If we've got 10% of cytosine, guanine will have 10%. We'll have 40% of cytosine because they're paired together, 30% of adenine, and 20% of thymine. Job's good 'un.